Well, I'm so glad I look like an actual mad scientist today. I get to talk about science. Hi, I'm Max, and at the time of filming this, I just left my job as a cell line engineer. I start a new one literally in like 4 days. It's all fine. But I wanted to actually finally sit down and talk to you about careers in STEM and what a cell line engineer actually is, because I've been meaning to do this since I got the job. So to understand what a cell line engineer is, you should probably know what a cell line is. So cell line essentially is a type of cell. So for example, you've got HeLa cells, which are cervical cells, you've got THP1 cells, which are blood cells, you've got HCT116s, which are colon cells. The list goes on. There are hundreds and hundreds and hundreds and hundreds of these. Cell lines are specifically designed so that they can be cultured for a long time without having any of their properties really change. So, for example, it's a really bad example, but go with it. If there was a cell line that had blue eyes and you cultured it for 20 years, it would still have blue eyes. That's probably one of the worst examples I could have thought of. They are a stable population, is what I'm talking about. So cell line engineering involves taking a cell line that already exists and genetically modifying it or editing it so that it will present certain traits or characteristics. There are so many fun ways to do this, but we used the CRISPR-Cas9 system, which currently everyone is talking about. Very, very, very basically, CRISPR-Cas9 acts as like genetic scissors. So we will send it in where we want it to go. It will do a little snip snip, so it will cut into the DNA, and we can do many things with this. Namely, we can use this to add genes in, and we can use this to take genes out. So adding genes in is called a knock-in, removing genes is called a knockout. For knockouts, typically you introduce the Cas9 system, it cuts, and you hope that it joins together weirdly in a way that results in a non-functioning gene. Whereas with knock-ins, you would attach something to the Cas9 system, which will then go and it will cut the DNA, and then hopefully it will insert what you've attached where you want it. So it adds in a gene. If you want to know more about the Cas9 system and knock-ins and knockouts, genuinely, the Wikipedia page is a good start because that will take you down any rabbit hole that you want to go. I'm not really interested in making like a feature-length film about the CRISPR-Cas9 system, what it can be like. That would take me forever, and I'm not a lecturer. But that's a good place to start because then you can go where you're interested. So check that out. And that's one of the only times I will ever recommend Wikipedia. Go and do your own research and click on the sources to learn. During my time as a cell line engineer, I did do a few rounds of CRISPR editing myself, but in my actual role, I was more towards the end of the pipeline, where cells had already been edited and they needed to grow up big and strong so they could go off and do their jobs. I was looking after the cells post-edit. After the editing event, every single cell in that plate, in that well, will have a different edit. And for the end product, we need a few billion cells, but each one of those cells has to have exactly the same edit. So I looked after each cell line when they were thousands of populations with different edits, and then shrunk those down into hundreds of populations with different edits. And then my team would decide which populations have the edit that we wanted, and then they would become less than 10, and eventually 1 or 2 cell lines. For each clone, there would be millions of cells. And I probably said billions the first time. I have billion on the brain, and we'll get to that. Essentially, I spent my days growing cells in plates and flasks, looking down microscopes, using big robots called liquid handlers for when there were thousands of populations. I don't have that kind of time. And doing all sorts of different tests on the cells, and in all honesty, becoming a bit too emotionally attached to some of them. I even got a tattoo of my favourite cell line, which you can't really see from there, but it says THP1. And I have billion on the brain because I cultured a billion THP1 cells got the tattoo to celebrate, and then I did a further 5 billion and ruined my shoulder. But it was so worth it. I get attached to these ones because I worked with them during my masters, and they're sort of known to be a little bit tricky, but I think they are babies and they are fine. They are very easy. It was one of the best days of my life when I got to hold all 1 billion THP1 cells in my hand at the same time. There's a photo of this on my Instagram. But yeah, I just had them all in a flask and I was just holding them. It was great. So why do we engineer these cell lines? If we already have existing cell lines, why are we engineering them? Well, there's so many reasons for this, but you can knock out a gene to see what not having that gene does to a cell. You can knock in a gene to see what having that gene. You can knock in an edit that's related to a disease so that you can see what that disease does to different cell types. You can knock in like drug targets. You can basically give a cell a genetic disease and then throw drugs on it and see if any of them improve the cell function. You can knock in a tag to see protein function in a variety of conditions. So if you have a protein related to disease, you can see what inflammation does to that protein, which is sort of what I did in my masters, a little tiny bit. You can create a disease associated variant and basically study that. You can throw drugs on it and see what happens. You can throw inflammation on it. You can give them communicable diseases. You can do so much. You can also like knock in genes that produce things. So for example, if you wanted to create antigens for vaccines, you could knock in a gene that would produce those antigens. We could make a cell line that leads to a better targeted treatment for a disease, or we could make the cell line that leads to better understanding of a disease, or that finds a disease associated gene that we didn't know before, which could then lead to better treatments, better targeting, better diagnosis, etc. It's sort of a well-known fact in the EDS circles that my type, hypermobile EDS, does not have a genetic variant, but we could find one using cell line engineering. Essentially, saving the world one cell line at a time. Jeez. How did I get this job? I had done my bachelor's in pharmacology and I did a master's by research, again in pharmacology. I've talked about my thesis somewhere up here. And I had all of like the lab skills that they were asking for, as well as like good teamwork, good leadership skills from coaching wheelchair basketball. So yeah, that's what a cell line engineer is, and sort of vaguely how to become one. Sorry it took so long. I hope that this has been interesting. If you have any questions, you can put them down in the comments, but I won't be making a follow-up video. I'll just answer the ones that I can. So, like this video, drop a comment because STEM, and see you in the next one. See ya!